Hello friends, in this video I'll talk about Sesame Floral biology, botany, economic importance and breeding prospects. Sesame, the scientific name is Sesame indicum, is one of the oldest crop in the world and is under cultivation in Asia for over 5000 years. The crop has early origins in East Africa and in India. India and China are the world's largest producers of sesame, followed by Myanmar, Sudan, Uganda, Nigeria, Pakistan, Tanzania, etc. The crop is highly drought tolerant, grows well in most kind of soils, regions and is well suited to different crop rotations. Distribution of sesame: India, China, Sudan, Mexico and all. India ranks first both in area and production of sesame in the world. Progenitors are Sesame angustifolium, Sesame radiatum and Sesame alatum. Belongs to family Pedaliaceae and chromosome number in diploid form is 26. It appears mostly in diploid form. Coming to importance, one of the oldest oil yielding crop. Oil content is 37 to 52%. Oil is rich in protein 20% as well as vitamin E. Used for edible purpose and also in soap manufacture. Oil is property of high stability, also used for culinary purpose and in bakery industry. Coming to economic importance, the sesame oil is rich source of edible oil. It contains 45 to 50% oil, has excellent stability due to the presence of natural antioxidants like sesamolin, sesamin and sesamol. Three antioxidants, potential antioxidants are sesamolin, sesamin and sesamol. Its grains may be eaten fried, mixed with sugar or in the form of sweet meals. Sesame oil is used as a cooking oil in southern India. The oil is also used for anointing the body, for manufacturing perfumed oils and for medicinal purpose. Sesame cake is a rich source of protein, carbohydrates and mineral nutrients like calcium and phosphorus. After oil extraction, the remaining meal contains about 35 to 50% protein and is rich in tryptophan and methionine. It is also a valuable and nutritious feed for milch cattle. In eastern and southern Africa, the leaves play an important role in the treatment of snake bites and malaria. In India and China, it is used in the treatments of cancer. Ash from burned stems is used as a medicinal salt. Oil is used to treat cough. As a solvent for certain kinds of oily injections like hormones, steroids, antibiotics. Sesame oil prevents sunburn, skin cancer and heals burns. Sesame enhances antioxidant activity of vitamin E. The tasty seeds are sprinkled on breads, cakes, cookies and candies and they are the source of valuable oil. The cake remaining after pressing or extraction makes an excellent livestock food and in times of famine is eaten by people. The seeds are eaten toasted or crushed and sweetened to make the Middle East candy known as halwa. Different names of sesame. In Kannada and in Tamil it is called as ellu. In Urdu, Sanskrit, Hindi it is til. In Telugu, Nuvulu and in Oriya, Rashiji. Coming to the nutritional value per 100 gram, the energy in 100 grams of sesame is 2372 kilojoules, that is equal to 567 kilocalorie. Carbohydrates are 26.04 grams, sugar 0.48 gram, dietary fiber around 17 gram, fat 48 gram and protein 17 gram. Origin and distribution. Tropical Africa is considered as primary center of origin. Secondary center is India. Sesame is distributed in centers of Asia Minor and Mediterranean region. Crop is cultivated in many countries including India and African countries. Systematic position. According to Hutchinson, the phyla is Angiospermae, subphyla Dicotyledone, division Metaclamidae, order Personales, family Pedaliaceae, genus Sesamum, and species Indica. Different species. Sesamum indicum is diploid, 26 chromosome number. Sesamum radiatum, chromosome number is 64. Sesamum prostratum, 32. Sesamum laciniatum, 32. Alatum, 26. Grandiflorum, 26. Occidental, 64. Morphology of Sesamum. It's a annual or perennial herb. Stem is erect, rigid, hairy, quadrangular, tall growing and branchy. Leaves are sessile and also petiolate, opposite towards bottom and alternate above, carry oil glands on the dorsal surface. Growth habit is indeterminate. Sesamum is an erect and annual herb. The plant is a simple or branched. The leaves are opposite below and alternate above. Coming to floral biology, flower opening is from top to bottom, that is basipetal succession in the plant bearing solitary flowers. The anthers dehisce from 2:30 to 4:30 am in the early morning and maximum dehiscence is between 3 am to 4 am. The flowers open between 5 to 8 am. Stigma is receptive at the same time as the dehiscence of anthers and remains so till 8 am. Hence the sesame is self-pollinated. Cross-pollination takes place by insects to a limited extent up to 5%. Sesamum have strong taproot system. The flowers are zygomorphic, solitary, occasionally 2 or 3 together, axillary, short pedicelled, borne on upper part of the stem or branches. Coming to floral morphology, flowers are solitary or in group, axillary, shortly pedicellate. Calyx is persistent or deciduous, gamosepalous of 5 lobes. Corolla is tubular, obliquely campanulate, gamopetalous, two-lipped, and ring of hairs towards base of stamens, variously colored. Androecium, the stamens are epipetalous, didynamous, filaments 4, slender, anther sacs are dorsifixed and dehisced longitudinally. Gynoecium, in which ovaries are superior, bicarpellary, two-celled, separated by false septum, two-chambered, ovules numerous, style is forked at the tip with stigmatic hairy lobes. Fruit is oblong or ovoid capsule with albuminous seeds. This is the floral morphology. The calyx, corolla, stigma, style, ovary are given here, dissected and shown clearly. Coming to anthesis and stigma receptivity. Sesamum comes to flowering 3 to 5 weeks after sowing. At that time, 2 to 3 flowers open in acropetal succession. But the flowering is in basipetal mode. Flower opening is between 5 to 8 am. Pollen viability is remain for few hours. Pollination. Natural cross-pollination takes place to the extent of 5%, that is through insects. Experiments show cross-pollination is up to 65%. It varies with location and different factors. The ovary is superior, bilocular, but may be subdivided by false internal walls. The fruit is a capsule, erect and oblong. Capsule contains numerous small ovate seeds. Testa may be smooth or reticulate and may be white, yellow, reddish, brown or black. The seed is dicotyledonous and albuminous. Here, short two linear bracts arise at the base of the pedicel just below the nectaries, which are shed when flowers mature. The nectaries are shown here just like small thorny appearance in the node region. At the base of single flower, two lateral flowers are observed as rudimentary buds. These are nectarial glands at the base, fully developed ones. Here it is shown as dissected flower of sesamum, the calyx portion. Calyx are short, velvety, narrow, acuminate and united at the base. 5 lobes are variable in size, lower are being longest and upper are the shortest. The flower are borne in the axil of leaves, either solitary or in groups of 2 or 3. These are already explained, but the pictorial representations are given here. This is different stages of flower bud development, and this stage is suitable in order to select it to emasculate for the hybridization technique. This is reproductive organs of sesamum flowers. The anthers, this is stigma, style and ovary, which are shown very clearly. This is a versatile attachment to anther and filament. Coming to hybridization, first selfing techniques. For selfing, bag the flowers for selfing to avoid natural crossing by insects. Another simple method of selfing is to tie a small soft cotton thread at the tip of the flowers about 12 to 16 hours prior to opening of the flower. Smearing of semi-solid clay. This is another method. Here, selfing can be done by smearing a speck of semi-solid clay on the upper portion of tubular petals of unopened flowers. The clay, while on drying, does not allow the tubular petals to open, and hence self-pollination is the rule. This method is cheap and less time consuming one. It is not effective during rainy season because during rainy season Fevicol may be, but the another method is during rainy season Fevicol may be applied on young flower bud to ensure selfing. Coming to crossing techniques, the first is hand emasculation and crossing. Flowers which are going to open on the next day morning are emasculated by removing the anthers with a pair of forceps through a short V-shaped slit made in the lower dilated portion of the corolla between 5 pm to 6 pm when the anthers are far below the stigma. Emasculated flower is bagged. Artificial pollination is affected on the next day morning and flower is bagged again. This is a process of emasculation, how it is done. The selection of flower buds. Next is soda straw method. Here in this emasculation technique, it is easy for crossing due to epipetalous nature of the stamens in sesamum. The flower bud which is expected to open in the next day morning is selected in the previous day evening between 3 pm to 6 pm and emasculated by just removing the corolla tube in which stamens are attached. Then the emasculated flower buds are covered with a piece of soda straw tube bent at the top in order to avoid contamination from foreign pollens. Coming to the breeding objectives, breeding for high yielding varieties tolerant to drought and high oil yield. Breeding white seeded varieties. Finest quality of oil is obtained from white seeded lines. Then development of mono-stemmed varieties. By this, more population per unit area and yield can be increased. Development of multi-capsule or axil and multi-carpellary varieties. Rice fallow varieties. For this, short duration sesamum is very important. Non-shattering varieties like African lines, then resistant to diseases like powdery mildew, Sesamum phyllodes. This can be transferred through wild species. Different varieties of Sesamum. Sesamum T7 has been released from US Bangalore, recommended for southern, central and eastern dry zone. Yield is 7 quintal per hectare. Other important varieties are Sesam RT46, Gauri, Rajeshwari, TKG 22. Breeding methods. First one is introduction, like African lines in order to avoid the shattering habit. Pure line selection, TMV6 from Andhra local. Hybridization and selection. In this intervarietal interspecific, for interspecific male sterile lines evolved by crossing with Sesamum malabaricum. Population improvement, polyploidy breeding, heterosis breeding. Heterosis breeding for epipetalous nature makes emasculation and crossing easier. Use of CMS lines is also being attempted. Embryo rescue techniques. Varieties Gauri, Madhavi, Rajeshwari, Shweta are developed through embryo rescue techniques. Coming to male sterility in Sesamum, occurrence of natural male sterility was first reported by Roy in 1931 in Sesamum. Male and female sterility was found by Kumar and Abraham, character being controlled by single recessive gene pair. Flowers are zygomorphic, solitary, borne on the upper part of the stem and branches. Coming to pests and diseases, leaf-eating caterpillar, that is Antagestra catalolmines, gall flies, Ashtandelia sesame. To control these, carbaryl 0.2% is effective. Then stem and root rot. Macrophomina fascioli is the causal organism. For this, seed treatment with Captan at the rate of 3 to 5 grams per kg of seed will be effective. Bacterial leaf spot caused by Pseudomonas sesame. For this, 3 sprays of agromycin at the rate of 1 to 100 ppm at 15 days interval. 3 to 4 sprays are effective. Then phyllodes, mycoplasma disease, then leaf curl, phosphomidon 0.05% or dimethoate 0.05% or dichlorovas 0.05%. This is floral diagram. Floral formula is calyx 5, corolla 5, androecium 4 and gynoecium 2. Coming to the wild species utilized in breeding program. First one is Sesamum alatum, that is having chromosome number 26, resistant to Sesamum phyllodes. The Sesamum alatum into Sesamum indicum cross is having dormancy. Sesamum malabaricum 2n=26 occurs in Travancore of Kerala. It is freely crossed with cultivated gingerly. Oil content is low as 32% and is utilized to induce male sterility in cultivated sesamum. Third wild species is Sesamum laciniatum. This has chromosome number 32, tolerant to phyllodes, drought and acid resistant, fertile auto-allopolyploid produced by crossing. that is Sesamum indicum into Sesamum laciniatum. The fourth wild species is Sesamum prostratum, occurs in Sesamum indica. This is tolerant to drought. Some of the references quoted in order to prepare the slides. Thank you. Hope you liked this video and don't forget to subscribe my channel. Thanks once again.